Thanks for joining us for another episode of Biotech and Life Sciences CEO Stories. Today, we're excited to have Dr. Josep Bassaganya-Riera as our guest, previously CEO of Landos Biopharma, which was purchased by AbbVie in March of 2024. And he's now CEO of the team at NImmune, where the original leadership team, which successfully founded, built, and took public Landos, is expanding upon the work that they did there in computational biology. Josep is the president of the NImmune Institute, a 501c3 nonprofit he founded in 2020 to carry on the original academic research and large-scale multidisciplinary projects in immunology and computational biology that had been the subject of his work at Virginia Tech. He's originally from just outside Barcelona, and he now resides in Blacksburg, Virginia, having previously been a professor at Virginia Tech. He earned his PhD in nutrition and immunology from Iowa State University. Josep, thank you so much for joining us.
Yeah, thanks for having me. My pleasure.
Your undergraduate work was in veterinary science. And I'm interested in terms of your personal journey, kind of what caused you to move from that into immunology. What caused the interest there?
Yeah, it's very interesting because as far as I can remember, perhaps when I was even 4 years old, I had made the decision, I had made my mind that I was going to become a veterinarian. And obviously, I ended up in vet school in Barcelona, in Autonomous University of Barcelona. I went through the entire degree. I graduated successfully. But in the midst of my vet school experience, I basically fell in love with immunology. Immunology touched every aspect of what we did in vet medicine, whether it's related to nutrition, it's how we heal after surgery, how we are able to fight microbes, viruses, bacteria, and at the vet school, I began to build a model in my mind of the immune system as a massively and dynamically interacting system with really tentacles extending to every part of our bodies and influencing health and disease. So basically, when I graduated, or a year before I graduated from the UAB, I started exploring opportunities to get deeper. And I didn't want to just explore immunology deeper as a single discipline. I realized during those early years, formation years, that the intersection of fields was very powerful from a problem-solving perspective. And so I was able to meet a gentleman who presented a year before I graduated, Dean Zimmerman, who was a professor at Iowa State. And he gave a really compelling presentation on the work he did with amino acid nutrition and immunology. And so I interacted with him. He told me, I'm towards the end of my career. I'm about to retire. I'm not sure I'm taking more PhD students, but I was able to convince him. And so I moved to Iowa, very cold, but a beautiful little town in the middle of the US. And then I pursued my PhD, and I took things in a different direction than Dean Zimmerman. His track record was, as I said, immunology and protein work. I took it into lipids and immunology. I did some seminal work on the immunomodulatory properties of conjugated linoleic acid, and that's how my research career in immunology started with a nutraceutical. In that process, we built established animal models of disease. We established the first pig model of inflammatory bowel disease, which later on would become very beneficial for the efforts at Landos and Immune at the NImmune Institute. And after that, I was offered a professor position at Virginia Tech. That's when I moved to Virginia.
So help me a little bit with that combination, immunology and lipids. Why did you go there?
The interaction between nutrition and immunology was at the core of how I had experienced during my years at vet school. I had been practicing during the summers. I was following veterinarians. I was treating cows. I was treating pigs. I was treating all kinds of animals. And I realized that the lipids in particular had the potential to have powerful anti-inflammatory effects. Now, what I had experienced in the vet school didn't have, the deep science that would justify the mechanism of action, but which it occurred. But I had seen, processes and results of treatments that justified looking at those more deeply. And for a long time, the anti-inflammatory effects of omega-3 fatty acids have been characterized, but I was interested in something more, something different, something that was a more of a new field of research. And I tackled a compound called conjugated linoleic acid, which is actually produced by certain bacteria in the stomachs of cows. And so it's basically a conversion of linoleic acid, which turns out to be pro-inflammatory, into a molecule or a combination of molecules that have powerful anti-inflammatory effects. And we characterized those, we elucidated the mechanism, and we got some nice understanding of that MOA. Now, that was not a drug, but some of the work we did on CLA helped us then advance into drug development, particularly in inflammatory bowel disease, because we built the tools, we built the animal models, we started to build the team that then would move into IBD drug development.
So there was a very direct link between your interest in veterinary science and immunology.
Absolutely.
Yeah.
I think that without that experience as a veterinarian in training, I would not have realized the depth and potential of understanding the immune system and harnessing its full potential for
Right.
therapeutic purposes.
Right. Joseph, I'm always interested in kind of the transition from academia to entrepreneurship. And I think you said when we spoke earlier that yours was driven in part by your relationship with your father and his successful business in Spain. Can you kind of sketch out that personal journey for us?
Yes, I think that this is an additional important pillar that shaped my next steps. In the 1980s, I was in elementary school in Spain. At that time, my dad was establishing his company that creates pulleys and rollers, all kinds of steel and aluminum parts. And he really started from scratch. He built that company from the ground up. I would see him, doing from the accounting to selecting the right machinery, to selecting the team members, to integrating each of the aspects. And while, um, my future was not connected to that kind of business. That was not my passion. My passion was, veterinary degree and then evolving into immunology. The kinds of steps and the approaches that you have to take to run a business were already embedded in me by this experience. And then the other aspect, science can happen in many different shapes and forms. And that experience and direct exposure to building a business from the ground up, which still exists these days, um, took me towards the kind of science that is more problem solving. And when I say problem solving, is as opposed to curiosity driven. Look, there's a place for both kinds of science. And there's people that are more attracted to one or the other. But personally, I'm attracted to, discovering mechanisms, validating mechanisms, but applying that new knowledge to solve important societal problems. And most of them in my line of research are related to public health, whether it's inflammatory bowel disease, you see Crohn's, infectious disease, metabolic diseases. So I think from that perspective, I was exposed to the inner workings of a business, and then the need for that business to be established in a very problem-solving way. And then the other aspect is how you accrue value in the long term, right? So in our current world, there's an expectation of fast turnaround of activities. When you build a business that is going to be there for 40, 50 years, you sometimes make decisions differently than when you build a business that you want to flip in 2 or 3 years. And look, there's nothing wrong about that. There's also a place for flipping, and we've had Landos acquired by AbbVie. We are very happy about that. But still, you can flip a business and still make decisions that accrue long-term value. Because if you've made those decisions, I think that in the long term, it's going to help the acquirer as well.
And therefore improve the value of the business when it's flipped.
Absolutely.
So back in, back in 2010, you led a group that was funded by NIH, which was authorized by Congress, to investigate ways in which iterative computational modeling could be used to model immune defenses. What did you learn from your work there, and how did it impact kind of the future course of your research?
Yeah, you're correct. So we were successful in securing one of 4 nationwide centers for modeling immunity for biodefense. As you pointed out, this was a congressionally appropriated program that was set up, I think, in response to the scares of the little powder envelopes that were sent to congressmen after 9/11. And the rationale behind that program, which I think is a very robust, thoughtful rationale, was to try to accelerate finding responses to these major infectious diseases scares. And so there was a realization that traditional methods by themselves were slow moving. You go through in vitro, then you do animal testing, then you go to the clinic, and every step is several months to several years. What modeling immunity for biodefense intended to accomplish was to build large-scale computational and mathematical models of the immune system that would be able to provide faster responses. You could run the simulations the similar way that DoD is running the war games to predict, okay, if you have parameter A, B, and C, what's going to be the outcome in that kind of conflict? If you have parameter X, Y, and Z, the outcome will be different. Therefore, you can organize your forces in this way or in that other way. So we were running simulations to understand if you have more regulatory responses, what's the outcome on this specific infection? If you have more effective responses, what's going to happen in that other infection? And I think that we made very significant progress. That was advanced computational modeling tied to supercomputing. We built a computer called Shadowfax, and that was able to compute trillions of agents at the same time. I believe that we developed the largest model of the immune system in 2012, 2013. And all that work that we did under modeling immunity for biodefense became one of the pillars of the next stages of our computational platforms. Nowadays, obviously, the work that we did in 2010, 2011 from a software development or software architecture perspective would be obsolete. But with the continuation of enhancement, refinement of the models, validation of the models, we have the Titan X platform, which has enabled us to identify novel mechanisms of action, identify novel targets, and advance in a very interesting way, I think increasing the probability of success novel therapeutics for autoimmune diseases and other conditions. So that work that started in 2010 has had a major impact in how we've accomplished things up to 2025.
What? So my understanding is that for each discipline, immunology, oncology, whatever it happens to be, for each one, there are now several models, several platforms of models that look to do the same thing, which is essentially predict the future. How is the Titan X platform different? What makes it special from others?
Yes. You are correct. There has been a proliferation of computational platforms that add efficiencies at each stage of scientific development. And in fact, we have a paper coming up that shows a comparison of Titan X versus several other platforms. Rightly, we'll be writing a press release connected to that paper soon. But I think one of the key elements that differentiates Titan X versus other platforms is the ability of our platform to integrate three very important pillars: the advanced computational modeling, the AI algorithms, and the bioinformatics. You may have several of those platforms that have an AI component, but don't have the computational modeling component. They don't have bioinformatics. They have other platforms that are very bioinformatics driven, but they don't have the AI algorithms and they don't have the advanced computational model. And so I believe that makes Titan X very complete, very unique, and it enhances significantly its power to make novel discoveries, particularly in immunology, but also in other fields. We built our computational capabilities to actually be disease agnostic. So we could use this same platform to ask questions that are related to epidemiology, and we would still get the right answers. Another important consideration about Titan X is, and that has been my bias, is that every prediction that the Titan X platform has provided, I force the team that has been successful in coming up with those predictions to run an experimental validation. In other words, we always went back to the lab and we made sure that whatever it predicted about behavior of the immune system or responses to treatment or target was validated experimentally. So I don't view Titan X as a replacement for the current methods of drug development or scientific discovery. What I view it, and that would be applicable not only to Titan X, but to many other platforms, they are layers of added operational efficiency. You can get things done faster. You can integrate millions or billions of data points and get answers faster. And that allows you then to go back to the lab and get that validation. Or if you come up that a response that says, oh, look, Titan X came up with this prediction, and unfortunately, that doesn't hold to be true in the lab, then you go back and you modify the parameters and see what's wrong in the model. Why was that prediction incorrect? And then through a model refinement process, you make sure that the next time the prediction is accurate. So I think that all those factors make Titan X actually very powerful.
Very interesting. So again, this past year, AbbVie acquired your predecessor company, Landos, and its lead clinical candidate, which was NX-13, a first-in-class daily oral, currently in phase 2 for UC. What was the process of drug development for NX-13, and how did it differ from traditional methods in a practical sense now? What were you able to shave from the development process?
So I think that the development of NX-13, and prior to that, the identification of NLRX1, the target for NX-13, which, by the way, now it's called AbbVie 113, is another validation and yet another success of the Titan X platform. Its computational capabilities were instrumental in identifying NLRX1. And there was something that resembled, there was a sense of deja vu in that the NLRX1 pathway is very similar to the LANCL2 pathway, which, as you may know, is the target that NImmune is currently developing with a drug currently in phase 3 clinical development. What's common in both pathways is both are gatekeepers in the intersection of immunity and metabolism. So they are very powerful proteins that enable, if you can reprogram what those proteins are doing in the broader context of immunity and metabolism, you can harness the energy of the cells, the amount of ATP produced, whether that ATP comes from glycolysis or from mitochondrial means, and you can coordinate that with the right kind of immune responses. In a nutshell, in both cases, what we are trying to do is shut down the excessive inflammatory responses that are associated with disease and pathology in ulcerative colitis, in Crohn's disease, in other autoimmune conditions, while enhancing regulatory T cells. Well, LANCL2 is slightly more effective at enhancing Tregs than NLRX1, but I'm very excited about both drugs, and I'm very happy that the next is now in great hands. AbbVie has demonstrated through its commercial capabilities, the success with Humira, that they can do very well, not only clinically, but also once the drug hits the NDA and starts to being commercialized. So I have high expectations for an X13 AbbVie-113, and I have greater expectations for Omilanco.
Great. Okay. Maybe we talk a little bit about kind of the business perspective for a second. Merck bought Prometheus for, I think, close to $11 billion last year. And they acquired with that not only the drug candidates for UC and Crohn's, but the advanced computational engine that supported their development. You chose, rather, to house the Titan X platform in Nemo, rather than sell it off to AbbVie. Why'd you do that?
So look, the Titan X platform is a platform that is currently available to NImmune Biopharma, a company that I'm leading on an exclusive basis. So the goal is to continue to develop the next generation of inflammation and immunology drugs under the NImmune Biopharma umbrella. It is the same company that is currently running programs in phase 2, NIM1324, in phase 3, Omilanco for ulcerative colitis and Crohn's disease. And I believe that the majority of my efforts for the foreseeable future are going to be connected to NImmune. Now, if what you are asking is why is the Titan X platform embedded within the NImmune Institute, and the answer is easy. It's the NImmune Institute is conducting the fundamental research. NImmune Institute is not conducting clinical trials. It's not in charge of doing IND enabling studies. It's doing the basic fundamental, although problem-solving research that will result in the next generation, the next wave of therapeutics. So it is absolutely fitting that the platform is available within the NImmune Institute. But because this platform is also used to identify novel biomarkers of response to treatment, and we've developed some biomarkers for Omilanco, which enable us to increase its placebo-adjusted rates for ulcerative colitis from about 27% to about 37%, and we are accomplishing something similar with NIM1324, then because of that, NImmune has licensed on an exclusive basis that capability to NImmune. And so it works really well. We can conduct the basic research under NImmune. We can do the more clinical development, and sometimes that requires slightly different teams, slightly different capabilities. So it's compartmentalized but there's deep collaboration between the entities, and it's working very smoothly.
So I read in a paper you wrote that you termed computational modeling in biology more than a technological advancement. You called it a paradigm shift. What do you think are the benefits in terms of target identification, drug discovery, and even clinical trials? Why is it a paradigm shift?
Yeah, you alluded just a few minutes ago to the fact that each field is starting to build its own advanced computational platforms. And I think that is a strength, but it can also be a weakness. And let me explain. It's a double-edged sword.
Okay.
And the reason is that the problems in the world are not compartmentalized to specific disciplines. And so if you build a platform that is only interested in some aspects of microbiology, another platform that is only interested and can only operate in certain aspects of cancer biology, it's going to miss opportunities. And the more transformative discoveries are going to be missed, are going to be lost in translation. What I said is that the Titan X, from the very beginning of its design, was intended to be agnostic. And another aspect that I'd like to add, which actually resonates really well with what I said earlier about that intersection of immunity and metabolism, is that we are convinced that the value of scientific discovery lies in the intersection between disciplines. So that's called transdisciplinary research, right? So the kind of research that we've been conducting, whether it's through Titan X or whether it's through Landos, through NImmune, transcends disciplines. We don't like to be labeled in a specific point. We may be very interested in the immune system, but we are not disregarding metabolic aspects. We are not disregarding aspects of computational biology. So I believe that that is one measure strength that has the potential to make significant advances. I believe that without that mindset and without those approaches, it would have been really hard to identify LINCL2 as a target for drug development, NLRX1 as a target for drug development, PLXDC2 as a target for drug development, LINCL3. All those are specific targets that we've discovered very efficiently, and then we've been able to validate in the lab, and we've been able to develop then therapeutics that bind and activate, or in some cases inhibit those targets, and we've been able to advance them to the clinic. The other aspect is probability of success. I mentioned that the way the original platform that was built under modeling immunity for biodefense was designed was to run simulations of the immune system to run what-if scenarios. Mm-hmm. Well, those what-if scenarios are still part of the architecture of how the system operates. And so you can run those what-if scenarios also in clinical trials. What happens if you have this population where you have maybe 50% active disease patients versus a population that has 70% active disease patients? What happens if you include 30% biologic failures? And what happens? And so if you run those scenarios and you have the outcome, then you are basically enhancing the probability of success in the end, because you've run computationally through enough failures, then you've been able to tweak the design to make it more likely to be successful when you have actually patients in the clinical trial. So those are some of the considerations: the probability of success, speed, the kinds of novel targets that are being identified, and the ability to leverage big data, whether those are large RNA-seq data sets or whether those are clinical trial repositories. In the end, what you add in the models, the value of that data defines the output of the models. The more valuable the data that you put in, the more likely that those predictions are going to be accurate.
I see a fair amount of pushback on the future and usefulness of computational methods. Not only here, just 2/3 of the top 20 pharmas have banned ChatGPT, and many in life sci call AI overrated, survey findings. I think this is from Endpoints back this spring. Do you have a comment on why, with all of the validity that you're finding and others, why is there this skepticism? Is it something about the seers of that skeptical view, or is it something about the limits of other computational methods other than Titan X, for example? They're not multidisciplinary enough or whatever.
That's a fair point. And when I evaluate the real intentions of an individual or an organization, I rarely stop by listening to what they have to say. I tend to focus on what are their acts. Actions are so much more predictive of what's behind an organization than words or press releases. And so you alluded to ChatGPT. Yeah, that's correct. That may be viewed as a distraction. But at the same time, you see each of the big pharma companies building their computational biology teams, building their computational models of the immune system or cancer biology, hiring computational biologists. And so look, if they want to ban these, these interesting tools, I would say that's noise. What I really focus is what are they doing? How are they positioning themselves to try to build the next wave of therapeutics in-house? Now, having said that, unfortunately, we all know that big pharma are not the greatest innovators. The reason they have to acquire biotechs is because innovation tends to thrive in a system that is less bureaucratic, that's more efficient, where there's the commitment of the team, where decisions can be made fast. It's like if you have a dinosaur versus a leopard, right? But the dinosaur ends up eating the leopard, but the leopard is much faster while it's free. And so what I would say, yes, there's a skepticism, and I've seen certain editorials and so on. But those kinds of transformative changes usually require the perspective of time. And so what I would argue is, let's look back 10 years from now at this conversation, and we will see whether there's any single drug that has been developed without taking into account AI, computational modeling, or bioinformatics methods. And if we find one, I'll say, chapeau, I was wrong. But I don't think we will.
Wow, that's quite a statement, Joseph. That's quite an optimistic and great statement for patients, at least, and I guess for drug companies as well. So you left Landos in 2021 to start NImmune, and the purpose there was to apply Titan X to the next wave of biomarker-driven immunoregulatory therapeutics for precision medicine. And then in 2023, NImmune reacquired certain late-stage clinical assets, such as Omalanacor, which is currently in phase 3 for UC, and NIM1324, which is in phase 2 for lupus and rheumatoid arthritis from Landos. Can you tell us more about those assets and in terms of the mechanism of action and their activation of LANCL2, which you mentioned previously, and kind of the timeline that you expect for each?
Yes, absolutely. As you pointed out, 2021, I stepped down in my role of president, chairman, and CEO of Landos to focus on the broader ecosystem. which included Animal Institute, NImmune, other aspects. When I stepped down, the company had a market cap of $600 million. And the new management took the company in a different direction. Um, and and so I focused on my next steps. By 2023, that different direction had resulted in an implosion of market cap of Landos. And so that provided an opportunity to have a conversation and basically articulate, and that there was a press release about that, an asset split transaction where Landos kept the earlier stage assets, including NX13, then now called AbbVie-113, and then I took over the more advanced assets, including Omilancor, NIM1324, and the entire LANCL2, LANCL3 platform. Just as a side note, more recently, I also helped Landos with the technical aspects of the AbbVie acquisition. So basically, had great conversations with the AbbVie team to show them the value of NLRX1, the value of NX13, and I was very glad that AbbVie and I supported once the implosion in value had occurred. I supported the acquisition by AbbVie. And so now we have the original Landos team that I have to say they are a team that I absolutely trust. They are committed. They are passionate. They've built this pipeline from ground zero. I remember the original idea in 2017, how we applied both computational and experimental methods to go from an idea to a product pipeline of 17 product candidates under Landos when we took the company public. This is the same team that wrote the S1, that made all value accruing decisions within Landos, and now they are committed to continue advancing the technology under NImmune. And they understand very well the mechanism of action of LANCL2. Yes, I mentioned it earlier. LANCL2 is another immunometabolic target like NLRX1. It resides in that intersection of immunity and metabolism. It's very important in regulatory T cells. As you may know, regulatory T cells are the anti-inflammatory arm of the immune system. And they are important in UC Crohn's, in lupus, rheumatoid arthritis, multiple sclerosis. If you have regulatory T cells that work well, you are capable of being protected from autoimmune diseases. Whereas if your regulatory T cells are dysfunctional, if they are weak, if they are unable to elicit their anti-inflammatory activities, then things start to fall apart. And you have lesions in the gut, can have lesions in the brain, you get all kinds of issues. So LANCL2 is actually very highly expressed in regulatory T cells. It's one of the top 3 proteins in regulatory T cells. There's even a paper, a proteomics paper from the Broad Institute, where they did really a fishing expedition, and they found LANCL2 one of the most highly expressed. But functionally, we've characterized LANCL2. We've developed drugs that bind and activate to LANCL2, and accordingly can enhance this regulatory T cell function and result in protection from Crohn's disease, from ulcerative colitis, from lupus, or rheumatoid arthritis. And more recently, we've discovered a novel angle of the LANCL2 mechanism of action. One of our colleagues, Dr. Lever, published in the Journal of Immunology that LANCL2 is actually very important in phagocytes, one type of macrophages, where the activation of the protein switches them from being pro-inflammatory to anti-inflammatory. Again, very aligned with the Treg part of the mechanism and instrumental in providing disease protection, preventing lesions, and so on. So we are excited about this mechanism of action. We are very excited about Omilancor. Omilancor, we published the complete and final data in October of 2023 in the annual meeting of American College of Gastroenterology. We've shown a placebo-adjusted rate in induction of about 27%, which is better than the Prometheus TL1A. We also have some comparisons of TL1As versus LANCL2 drugs, which we'll be publishing shortly. And Omilancor is pushing forward in terms of timeline. Our goal is to have top-line results for Omilancor in ulcerative colitis by 2027. And the FDA approved the phase 3 protocols on two independent, well-controlled trials, one with about 600 patients, another with about 700 patients. And Omilancor is advancing well. NIM 1324, we just had a publication accepted on the results of the phase 1 clinical trial showing outstanding safety and tolerability. It has a slightly different PK profile than Omilancor, and it's showing very powerful therapeutic activities in lupus, in rheumatoid arthritis, and a broad range of other autoimmune diseases, which we have not disclosed, but have the potential to allow hits with NDAs almost on a yearly basis, in similar way to what AbbVie did for Humira.
So I saw an article about a study at Hunter College that said that Omilancor has some potential in Alzheimer's as well. So kind of a strategic question. All the data, Titan X, LANCL2, applicable to all these different diseases. So you must feel like a kid in a candy store. You can do anything. How do you focus and focus the team on specific diseases when you have so many opportunities, and yet we're in an age of limited capital availability for biotech? How do you focus the team?
I think this is one of the most important responsibilities of a CEO, having the team focused on a laser thin way on what's the most important item at hand. And I can tell you that our team at Immune is focused right now on ulcerative colitis and Crohn's disease. This is the top indication. I was very excited. I was not part of the design of the Hunter College work. I saw it tangentially. I was happy to see that there was a CNS, a neurodegenerative disease indication that we had not even started to dig into, where there was validation for LANCL2. Now, we are not at the point where we are going to stop the work that we are doing with IBD and redirect the efforts towards Alzheimer's disease, but we take note. And the composition of matter is the composition of matter. So if Omilancor or NIM 1324 show efficacy in Alzheimer's disease, I think this is just an additional amount of value that has been accrued indirectly through the efforts of outstanding scientists in Hunter College. But we remain focused on IBD. NImmune Biopharma is an autoimmune disease company, but our current focus is ulcerative colitis and Crohn's disease. This is the top priority, whether it is the clinical trial work, the better understanding of the mechanism, or the validation of the biomarkers of response to treatment, which will ultimately increase the efficacy and will allow us to have better negotiations with the payers when we get to the reimbursement strategies.
Joseph, you developed a therapeutic that was important enough to be acquired by a major pharma after having already experienced an IPO. So I wonder, to a founder just starting out, what's your best advice for that new founder?
Yes, that's a really tough question. I think that and look, what I'm about to say is it could be viewed as controversial, but I'm going to say it nonetheless. I think that over the years, I've been focusing on providing the kind of leadership that is grounded on, authenticity. I follow my gut, and I like to get things done. But the most important advice for a new funder is to be aware that this is a world where the environment, unfortunately, is not founder friendly. And so, there's many funds that will say, and maybe that's very attractive for the LPs, for some subset of LPs, that they are very founder friendly. The reality is that many are not. And so being able to see through what is really founder friendly versus what is not founder friendly is going to make a difference for his ability to be able to implement his vision, for his ability to be able to take whatever drugs, whatever new transformative treatment, all the way to the finish line, and ultimately change the life of patients. If you look at the track record of many funds in developing drugs that make it to the patients, it's very limited. The process tends to be jumping, transferring the risk to the next one. And so are there true organizations that think in the way that maybe Warren Buffett has been thinking? I know that he doesn't invest in biotech, but nonetheless, the kinds of comments he makes, he wants to buy the companies that 30 years from now are going to make a difference. That strategy in the long term, Berkshire Hathaway has demonstrated, has been very successful. So my advice to founders is to be able to see through the noise and identify the right partners that, in spite of what they say, their actions demonstrate the truth or not truth of those statements.
Okay, very good. Joseph, thank you so much for taking some of your time to share the details of your work with us today. I think we all share your excitement about the prospects for AI and computational biology, and we're developing more accurate targets and more productive clinical trials. Thank you so much.
Thank you very much. It was a pleasure talking to you.
Thanks for tuning in to the Life Sciences and Biotech podcast. We'll see you in the next episode. The information contained in this website and podcast are purely informational and not considered investment recommendations. Tim Doherty's participation in Biotech Insights is separate and apart from his role as an investment advisor representative. Nothing contained herein can be construed as a recommendation or endorsement of any of the companies discussed. Tim Doherty also has no financial affiliation with any of the companies mentioned in this communication. Tim Doherty makes no representation that the information contained in this material is accurate and is under no obligation to update this information as changes occur.